So there, we're recording now. Um, and yeah, today I'm talking about the ecological niches of bats. A bit about my background. I've been working with bats for a little over 10 years now. I started in my undergrad doing an honors thesis project looking at isotope ecology of one community of bats from northern Belize. And then I carried that on to the University of Waterloo, where I did my master's, also with Dr. Hugh Broaders. You sort of broadening out to the isotope ecology of neotropical bats, looking at niche partitioning, and how these animals sort of interact with each other in their environment. I've recently finished my PhD at the University of Toronto with Dr. Ken Welch. Um, where I was looking at this across various different contexts and various landscapes, examining the isotopic niches of bats from the individual level all the way up to the community level. So I've been doing this for some time, and now with Dr. Beth Clare, trying to sort of expand out this toolkit beyond the isotope ecology end of things and looking more now at what bats are actually eating through molecular analysis and also learning some handy dandy eDNA stuff. So hopefully I'll have some different things to talk about uh, if we do this again at some point in the future. So some points that we probably all know about being in GBatnet and coming to talk about bats is that bats are very diverse. There's 1,500 species of bats now described, probably more than that out there. This diversity is both in terms of taxonomic diversity, where they form these very diverse communities, particularly in the neotropics. But there's also a lot of other types of diversity, where there's morphological diversity, um, diversity in the way that bats echolocate, in the shape of their wings, in terms of their physiology, even how they process sugars, for example. And this is also captured in their trophic diversity. So this is just sort of a little exercise of these are all faces of bats from a community in Costa Rica where we work. Uh, and I wanted to just sort of box them into their trophic guilds. What would we expect these bats to be based on the literature and based on sort of general thoughts of their ecology? So at the top here we have our fruit-eating bats, generally thought of as frugivores, short, broad faces, good crushing jaws, things like that. In the middle here we have our nectar-feeding bats, these elongated rostra, long tongues, reduced dentition. At the bottom here are insectivores. These are gleaning insectivores, so a little untraditional from our aerial hawkers, but these are bats that will predominantly eat insects. And then over here we have our carnivorous frog-eating bat and Phyllostomus cystatus, which is also sometimes described as an omnivore, but I'll get into that later on in the talk. So this is all a neat little picture, and this is how bats fit together in this community. But then when we start to sort of pull at the threads, it really begins to break down. So we have two different bats in the same genus that are in different trophic guilds. As I said, both probably better described as omnivores. Are fruit-eating bats also eat probably a lot of nectar and vice versa to some extent. And all of these bats, in one way or another, will eat insects, at least for a certain time of year. So this was sort of the core of my PhD dissertation, which brought me to these three sort of key questions within this context of the ecological niche. So what is the role of trophic flexibility in bats that are otherwise specialized? How do similar species partition their diets and their dietary niche when they occupy the same trophic guilds? And how do so many different similar species coexist within the same faunas? So as I'm going through today, I'll be talking a bit about my work, a bit about other people's work. But this is sort of the core of what I'm interested in and was interested in through my PhD and continue to be in building my own research program. So I've said this word a few times, and this being my field for dummies, I thought I'd bring it back to the basics. So what is a niche? This was a bar in Halifax. I'm not talking about that. This is a architectural niche where you would put a statue or something. Also not talking about that. And as they are typically wrong about a lot of things, the American pronunciation of niche is also there's no T in the word niche. We'll use the British Canadian. It's better. What is a niche? In order to really sort of talk about what a niche is, I need to go back and talk about these niche concepts. So there are a few different definitions of the ecological niche, and these have progressed with our understanding of ecology over time. So the first one was Grinnell in 1917. We describe it now as the Grinnellian niche, which is that the niche of a species is encompassed by the habitat in which it's found and its behavior. So relating the behavior of an organism to its environment. And that's quite broad, which brought Elton in 1927 to really sort of try to cement that more in the place. So, and also in relation to others within the same communities. So it's the place of an organism in its relation to its food and its enemies. So, sort of building on this trophic structure idea, how organisms interact with each other rather than with their environment. Which then we come to Hutchinson, which was in 1957. And this is still sort of the current understanding of what an ecological niche is. And the Hutchinsonian niche is defined as the n-dimensional hypervolume, which defines all axes in which an organism exists in its environment. So that's a bit jargony. What is an n-dimensional hypervolume? So if we think about a two-dimensional grid, so there's an x-axis and a y-axis, that is two of n dimensions. If we add a z-axis, that becomes three of n dimensions, and so on and so forth. So as we look more deeply at an organism, they exist within all of these different dimensions. They exist within space, within time, uh, within their interactions with their environment, with each other, with what they eat, with where they live, with where they travel. So, as we as ecologists think about this n-dimensional hypervolume, the more dimensions that we're able to examine, the better our resolution is of what the organism actually is doing in its environment. Some of these are very difficult to measure, obviously. Some of these are much easier to measure. Um, and as we're sort of going through this, the real thing is there's always more out there. So these n dimensions in theory extend, as many dimensions as there are as we're able to actually conceive of. So this is a very pared back discussion, and we typically will revert to thinking about this only at the population level. But there's also this lovely paper that came out in 2022 thinking about it at the individual level as well. So individuals have their own n-dimensional hypervolumes around them within this population. I'm not really going to touch on this today. I have a paper in the works about this, about a community of bats where we have permanently tagged individuals. But just to know that this is not all at the population level, this is a good place to start if you want to check that out. So, as I said, some of these dimensions are easier to measure than others. Movement, for example, we can track bats while they're foraging, examine their both home range and potentially migratory range using tools like MODIS. Uh, Diet, we typically will look at studies in diet and ecology, so we can characterize what these animals eat. We can characterize what they're eating relative to one another. But quantifying it is often difficult depending on who you're talking about. So we can look at fecal analysis or stomach contents, um, but actually, those are tend to be more presence absence studies rather than sort of the proportion of diet. Other things we typically think about when we're talking about the ecological niche is roosting ecology, their environment, so the temperatures or humidity that they can tolerate. Um, as I said, this is n-dimensional, so I'm just going to sort of talk about these two, and I will do that within the context of stable isotope analysis. So I know that there was a My Field for Dummies talk on stable isotope analysis. I would encourage you guys to check that out on the YouTube channel. But just sort of very briefly here, so the two most common, I would say, isotopes that people use in studies of ecology are carbon, denoted here as delta 13C, and nitrogen, delta 15N. So these are the ratios of heavy to light isotopes of each of these elements in animal tissues. in these cases. And when they're used together, we can develop a two-dimensional isotopic niche. So this is not quite a dietary niche. It gives you a bit more information about spatial foraging and diet. So it's a combined factor here. So carbon here on the x-axis will typically be conserved in animal tissues from the original source of carbon in their environment. So C4 photosynthetic plants have very different carbon signature than C3 photosynthetic plants, for example. Marine systems have a very different signature from aquatic systems, which would sometimes be way down here if there's a lot of methane in there. Um, and even within C3 plants, there's a canopy effect. So in tropical forest systems, uh, fruits that grow higher up in the canopy tend to have higher carbon than those that grow closer to the forest floor. So you can sort of tell where an animal's foraging rather than relevant to one another. Nitrogen increases with trophic level at a rate of about 2.5 parts per mil per trophic step, 2.5 to 4, not a hard rule. And then using these two things together, we're able to generate two-dimensional isotopic niches, as I was saying. Um, there are a lot of other different stable isotopes that we're able to use, and they're useful in other contexts as well. So we can use these to also study migration in bats. If we have good isotopic maps of regions of whole continents, even, we're able to really, and we can catch a bat on the landscape, clip its fur, and analyze it for these isotopes, we're able to actually match it up to figure out where within their environment, or where within the continent this animal had come from. So, carbon, nitrogen, as I mentioned, delta deuterium, hydrogen, oxygen, strontium, sulfur. There's a lot of different options for this type of analysis. And this is a great paper as a starting point to check that out if you're interested. So, what I also wanted to do was talk about how we use these metrics to examine shared niches across various ranges, and how do animals within the same communities partition resources if we're capturing them foraging together. So, the I'll talk very briefly sort of about this first point. I was the first measurable niche dimension that I brought up earlier, which was the movement of bats. So when we think about bats in terms of their spatial use, there's sort of these two different categories. There's nightly movements, where bats go from their day roosts, typically to socialize or feed or explore their immediate surroundings. And then there's also seasonal migrations. Which I don't really have time to get into, but it's another thing to consider. So foraging ranges, these are all three species of neotropical bats. Um, a lot of the time, they can be quite close to home. Some bats have quite a high site fidelity. So Carolia perspicillata, fruit-eating bat, they're typically will move, or at least from the studies that I've seen, about 500 meters from their roosts. So they have pretty much everything they need in their immediate environment. Artibius literatus, approximated about 1.3 kilometers is their foraging range. So they'll fly out, look for figs or other fruits and return to a roost. Glossophaga soracina, in this case, this study quote said that their approximate home range is about 3.1 kilometers in any given direction. So they fly, even though they're a much smaller animal, they fly about double the distance of a much bigger bat in Artibius literatus. Um, but they're also capable of doing these long distance movements. So even though we can think about their spatial niche as being sort of within these bounds, they often will, or occasionally will, step beyond that. So this is a record of Artibius literatus, really sort of a fortunate record where they were permanently marked in Brazil. And then they were being recaptured hundreds of kilometers away. So even though the typical home range size of Artibius literatus was about 1.3 kilometers, as I said, this bat was caught 113 kilometers away from where it was originally found. So when we think about this in terms of their foraging area, that this aspect of their niche, we really need to consider how flexible these animals can be in terms of their movements. So this is a case of bat where probably been discussed many times in these talks, Leptonictus yerba where these bats live in a central roost, but their foraging range on any given night is massive by comparison. Um, so these bats will roost in caves along the coast in Mexico and then travel hundreds of kilometers, or over 100 kilometers in a night in one direction to go feed in columnar cacti. Um, these in and again, just sort of when I was talking about the niche of the individual, they have very consistent preferences. So here's a central cave where all these bats were tagged with GPS tags, and then night after night, each individual would fly out to a patch where they knew there were columnar cacti and they were preferring to feed. This bat, for example, here flew over 100 kilometers each direction just to find these particular cacti, um, and returned home over 200 kilometers round trip in a night. So when we think about the foraging niche, the spatial niche of these bats, um, it's sometimes really context dependent and really mind-boggling, considering that, all these bats share a space together, but then spread out so vastly across the landscape. Um, Using carbon and nitrogen stable isotope analysis, we're able to sort of see this, where in fission fusion societies, this is a study from Maritime Canada. Uh, in the summer, there's very high foraging site fidelity where bats are more similar within their own populations. They're feeding and growing hair uh, in particular areas. And then at roosting, at autumn swarming sites, when they come back together, we're able to still sort of determine where these bats have come from. So the the context is really it's all very context dependent. Um, Looking back at these same species I was talking about earlier, Artibius littoratus, Carolia perspicillata, at a in a the Atlantic forest of Brazil, it's also context dependent in a smaller area as well. So this is just a few fragments near a contiguous forest outside Rio de Janeiro. And bats that are found within the contiguous forest have considerably different diets from those that are found out on the forest fragments. So sometimes they can be locked in rather than, have these sort of very broad foraging ranges. I mentioned roosting as well as something to consider. And that's sort of in this context of there are various different foraging or roosting types. We have bats that roost on bark or in foliage or in these giant caves. But then when they roost together, what happens when these bats share a common roost? So if we have these really diverse communities like we have in the neotropics, um, and one dimension of this n-dimensional niche is shared across the entire community broadly, so we take that one away. How are bats partitioning resources? How do they coexist? So in order to tell this story, this is some of my own work that brought me to Jamaica. I grew up in Toronto and very fortunate in Toronto to have the Royal Ontario Museum. And within that museum is the Bat Cave, which is a model of St. Clair Cave in Jamaica. So this had always sort of captured my attention. And, seeing this cave where there's these distinct roosting areas where the fruit-eating bats roost apart from the insect-eating bats, but within this confined space. Um, I really wanted to go there. So through my PhD, I uh, pushed, and Ken's in the chat, and I always said that Jamaica was a great place to go to do research. So we actually went and we caught bats at St. Clair and 30 other caves around Jamaica to really try to understand how these bats are partitioning when they share this common roost space. How do they separate out in this isotopic niche space when their habitat is so similar? As many of you know as well, Jamaica was just hit by their worst hurricane ever. These are some photos from Cockpit Country, which is the largest protected forest in Jamaica. and it's really devastating. I'm just gonna throw up this here, just in case anyone was looking for a place to donate to. Um, but I was supposed to be in Jamaica in uh, 6 days, and that trip is not happening now as a result of the damage caused by the hurricane. The Yeah, anyways, it's sort of mind-boggling to think about how much has been lost there, but we're hopeful to keep this research going and see what the effects of this has been on the bats. Anyways, I digress. Back to niche occupancy. So this is a one of the chapters from my dissertation that's currently in review at in a journal. But just to sort of give you some background on the bats of Jamaica beyond my personal connections, um, there are 1,000 documented caves in Jamaica. About 16% of those are the substantial bat roosts. So I was talking a lot about St. Clair. This is one of many caves where there's these communities of hundreds of thousands of bats. There's 21 different species. About 15 of them can be found in caves or obligate cave roosters. And they span all sorts of different species from various families, a lot of Phyllostomids, Mormupidae, Natalidae. They're all over the place. These caves across the island vary in size and complexity, number of entrances, and interestingly, in species composition as well. So we went to Windsor Cave, which was the those photos I showed at the top of all the stripped and broken trees. That's just outside Windsor Cave. That was the most speciose cave on the island by our estimation, with 16 different species. There's caves where there's only Artibius. The least speciose that we visited was Vauxhall Cave, where we saw a few Artibius, and there's a record of Nyctinomops. Um, but across the board, these caves vary a lot. And the central question that I wanted to address with this was that how do so many similar species coexist in these very diverse sites, and what happens at these less diverse sites by comparison? I was inspired when I found this paper by Rohit Chakravarty et al., which looked at how species richness across elevational gradients impacts niche packing and niche expansion. So essentially, what they were looking at was that at high elevations where you find fewer species, species in this niche space tend to spread out more. Whereas when they are at lower elevations and it's more speciose, these niches pack together into sort of a confined niche area. So niche space, how does that exactly work? Where do these concepts come from? Um, Largely, this comes from MacArthur, who was one of Hutchinson's students, actually. And these concepts of niche packing and niche expansion originate from these ideas uh, of coexistence. So, how do animals, when they're in more diverse sites, exist together? Niche packing can occur through two potential mechanisms. So the first one that we were examining was niche packing through specialization. So as there you get to sites where there are more species, niches sort of collapse down. Animals become more specialized when they're faced with greater competition. So I'm really good at this. I'm going to get better at this, and then that way I don't have to share my resources with other people, or they can increase niche packing with increasing overlap. So, in a situation where resources are potentially not limiting, or where there's less competition, or even in cases where there's no other options, overlap may increase, uh, which in our context is indicative of resource sharing. So bats will share more in terms of what they're eating, what they're doing in the environment. In our cases with these caves, we know they're already sharing their roosts. So how much can they potentially share outside? To do this, we went to Jamaica. As I said, we set up herp traps in front of caves, visited 30 different sites around the island. And then at these white squares, I have isotopic data from carbon, nitrogen, isotopic data from hair, from these bats. Uh, in the process, I just wanted to plug this for a second. We did find two new roosts of the critically endangered Jamaican flower bat. These bats, prior to my 2023 visit to Jamaica, were only known from a single roost, and there were about it was estimated to be less than 500 individuals remaining at the single roost. But then, yeah, traveled down there working with Ronnie Hall and Damian White at the University of the West Indies, and we caught them at two new sites, and they were pregnant females, and that was awesome and great hope for the species. Anyways, so I have isotopic data from all these bats from 13 unique sites. There are about 12 of the 21 species are represented in our data set. And I used linear models to test relationships between isotopic data and species richness. And the isotopic data I was looking at were three different things. So I was using mean overlap. So this is estimated using a package called Niche Rover, which if you're interested, I can tell you about later. Um, Mean overlap within those communities to look at that niche packing through increasing overlap. Mean niche breadth, which is estimated as the Bayesian standard ellipse area. Again, I can tell you about this in more detail later. Uh, and that was to look at specialization. So if there's a relationship between niche breadth, how broad an animal's isotopic niche is relative to one another across the communities, and then also mean nearest neighbor distance, which is one of these community-wide measures of isotopic structure. And I looked at this across three different scales of what we would consider a community. Within family, the two most represented families, Phylostomids and Mormupids. Within trophic guild, Nectarivores and Insectivores, and across the full fauna. Um, so what we found was that yes, there is a uh, negative relationship between species richness and nearest neighbor distance. So bats are within these communities, in more diverse communities, they're packed more into sort of a defined community niche space. So there's greater niche packing when we have more speciose cave roosts, which is a clear support for our niche packing hypothesis. Where we're looking at niche breadth to see if this is through specialization, there's no real relationship within Phylostomidae. Within Mormupidae, we actually see a positive relationship, though it's non-significant, where at sites where there were more species of Mormupids, the niche breadth actually expanded. So that's contrary to that niche packing through specialization. And same thing within guild. Our insectivores, there's this positive, though non-significant relationship, and our nectarivores is pretty much this flat line. So we can say that niche packing through specialization is likely not occurring. And then looking at this again through overlap, we see this positive relationship for Mormupids. This time it is significant, where at sites where all four extant species of Mormupidae occur on Jamaica, there's significantly more overlap among them. Though this is not the case for Phylostomids. Again, within guilds, insectivores, there's no significant relationship, though it's slightly positive, but our Phylostomid nectarivores, there is the significant association. So at sites where there's greater species richness, there is also greater overlap. So this supports this idea that niche packing is through increasing overlap, which might indicate that they're sharing more resources, they're potentially competing more. Um, and it really sort of puts this into a context of how much can they share within the landscape. So the next steps with this are largely uh, thinking about it in terms of what their foraging space. So we're going to try to be examining more of the bats on the landscape. Um, and thinking about more in terms of how much can they actually tolerate. So even in the situation of the hurricane, where so many of the resources have been destroyed, um, trying to examine how these bats are shared, partitioning the resources, how they're competing, and how these communities exist over time. So what happens when bats share both foraging space, roosting space, and diet? How do they partition their niches in diverse faunas when there's so many of these end dimensions overlap? And this was another point that I was thinking about a lot through my PhD. Uh, and one of my key hypotheses was that this is done through omnivory. So we know that bats are quite flexible in what they can do, but in terms of our how we think about bats, it's typically we put them into boxes. So these were two studies that were done at Laminae, Belize, which is a very well-studied fauna. It's been basically since the 1990s, I think, was the first bat trip there by Fiona Reed and Brock Fenton. Um, and it has been an excellent place to study these sort of long-term changes in forest and niche ecology. But we really sort of set out at first to just define what these bats are doing. So I, through my bachelor's and master's, was looking at this in this isotopic context, trying to examine community structure. So each one of these ellipses is a population's isotopic niche. And trying to see how they overlapped with one another, how they spatially arrange within this community's isotopic niche space. And what I found was that a lot of the time there's significant overlap between bats that we typically think of as one thing or another. So our nectarivore here in purple, the time was Glossophaga sorcina, now it's Glossophaga mutica, overlaps a lot with both our frugivores and our insectivores in this isotopic context. Our carnivores overlap a lot with our insectivores. So why is this? What's going on? Are these animals actually competing with one another? Are they sharing resources? Um, we know that they're foraging together, so how do they all coexist in this space? Melissa and Gala then a few years later published this paper looking at the actual dietary components of what these bats were doing. And there were some surprising things, but things that were supported by the isotopic data. Cryptopterus ericeus, large carnivorous bat, was eating plant matter and insects largely, very little in the way of actual vertebrates. Pteranodus mesoamericanus, this smaller, more mupid bat, was eating plant matter as well. So it was sharing resources with our frugivores to some extent. And our fruit-eating bat, Jamaican fruit-eating bat, Artibius jamaicensis, was eating a substantial amount of beetles, a lot of Coleoptera. So this sort of brings me back to this idea of dietary guilds, and I'll go on a bit of a diatribe for the last 15 minutes here. Um, and when we think about dietary guilds of bats, there's typically six that we can put bats into: frugivores, nectarivores, insectivores, piscivores, carnivores, and sanguinivores, which is a mouthful. And I'm gonna sort of get into why I don't think this is a good idea. But if you want to read about that as well, there's this book chapter in the Natural History of Bat Foraging, which I would recommend checking out. So I want to go through this one guild at a time, and just sort of contravene your expectations of what is the dietary niche of a bat. Um, the first one is our sanguinivores a guild, our three species of vampire bats a guild. This is the one where I'll go back on myself and say that yes, indeed, they are probably just a guild on their own. These are highly specialized animals that just eat blood. But let's get deeper into it. So our insectivores a guild. When we think of an insectivorous bat, there's many different foraging modes. There's many different ways that insectivorous bats go after insects. And insects themselves are very diverse. So what are they eating broadly? If we consider diet as one of these n dimensions of the n-dimensional hypervolume Hutchinsonian niche, uh, how do these bats partition within, say, a diverse community where there's 20 different insectivorous bats that all live together? So Denzinger and Schnitzler published these functional foraging groups, which is largely based on their ecology, sorry, their echolocation strategy, and suggested these six different categories that we can split up this sort of monolithic insectivores into. They're bats that are open space foragers. These are like largely like Melosidae, free-tailed bats that will forage high up in open spaces. Edge space aerial foragers, so hawkers that occupy the edges of forests. Edge space trawlers, bats that will trawl uh, insects or things off the surface of the water or off the surface of plants. Uh, Narrow space flutter detectors, narrow space active gleaners, and narrow space passive gleaners. They also suggested a seventh category here that encompasses fruit and nectar feeding bats as well, but I'll get into why that's kind of problematic later on. So I'll just give a couple of examples of why this niche concept of putting bats into guilds doesn't quite work out. So Anthrazoos pallidus, the pallid bat, very well studied bat, present here in Canada. Um, and they're found to in mostly arid landscapes, and they're believed to be scorpion or centipede specialists for a long time in literature, mostly thanks to photos like this from Merlin Tuttle. Um, we know that this is not the case, though. So, Fred Frick et al. In 2009 described this unique habit of Anthrazoos pallidus, where they were found covered in pollen. And they set out to, there have been several studies on this since, but they've examined this and the pallid bats love sugar water. So they will go out and specifically forage for nectar in I believe this was in the Sonoran Desert. So they'll fly around seeking this out, even though these are bats that are supposed to be eating scorpions, centipedes. These are insectivores that are eating nectar. They're performing as something that we think of typically as a nectarivore. Um, Really, they were intentionally going after this. Only 2% of their flower visits involved interactions with insects. So they were actively seeking nectar as their for their diet. Insectivores do often eat a lot more than just insects. So please consider this when you're examining this concept of the dietary niche in in these types of communities. So going on with from the insectivore that eats nectar, let's talk about nectar bats for a second. So these are highly specialized animals. They have unique tongues, unique rostra, skull shapes, the hovering flight that they're able to do in front of inflorescences. They're built to eat this floral nectar. Um, but when we consider how diverse they are, or how morphologically diverse they can do a lot of other things. So these are all photos by Jose Gabriel Martinez Fonseca, who gave the talk last week, I believe. Um, and these are all bats that we catch at this community in Costa Rica. Um, Very quickly, just I don't have any ability to take a poll here, but if you just think about who is the least specialized among these bats, who would be the bats that would eat the most nectar, who would be the bats that eat the least nectar. I just want to highlight two of them here. So Phyllostomus discolor and Lonca robusta. So Phyllostomus discolor, they're typically described, they're really sort of, they're mixed up when we think about them in the literature. They've been described in a whole bunch of different ways. So sometimes they've been described as insectivores, or they can be described as insectivores. Fleming et al. Reported that 99% of their stomach content was from insects. So if you were just looking at that one data point, you'd call this an insectivorous bat. Are they omnivores? Uh, Other sources have reported vertebrates in their diet, fruit, pollen, um, or are they nectarivores? 70% of their diet reportedly comes from pollen and nectar in some faunas. So when we think about the dietary guild of this bat and the dietary niche of this bat, how wide does that extend, and is that entirely context dependent? We need more studies on this, to be honest. Lonca robusta, this is sort of more typically the shape of what you would think of as a nectar bat. Um, Again, maybe could be considered insectivores depending on what you're looking at in the literature. So 90% of their stomach content was insect in that same Fleming study. Um, this might have been a case where this was seasonal. So when we think about the dietary niche of these animals, it needs to extend beyond a single point in time. So there are records of bats switching almost entirely into insects like Anora jeffrey and Corollia Glossophaga mutica. Uh, and we need to remember that these nectar feeding bats are extremely good at being insectivores as well. So when we're thinking about nectar feeding bats, just remember that they eat a lot more than just sugar. Are frugivores a guild? So fruit bats typically are well described. It typically says fruit bat in their common name. Um, we have everything from these very large pteropods to phylostomids in the neotropics. Uh, and they are out most of the night, usually eating fruit. But when you just eat fruit, there's often a lot of things missing from an all fruit diet. So there's a lot of different essential nutrients that are lacking in many different fruits. Some fruits that are better for filling some of these needs than others. For example, calcium is largely filled if you're eating a lot of figs. But there's still things that you're not getting. So when you're out seeking, if it's a particularly metabolically intensive period, say that you're in the middle of reproducing, you're pregnant, you're lactating, you need more nutrients. Um, these are two very common neotropical fruit bats, Corollia perspicillata and Artibius litoratus. And both of these bats are well documented to eat a lot of insects as well. Corollia perspicillata a little more so, but more and more people are finding records or finding evidence that these bats are eating insects. This is often unaccounted for, though, because of foraging mode. So Corollia will actively spit out the exoskeletons of bats, sorry, of insects. Uh, and that's why in these fecal if we were just looking into fecal components or stomach contents, we wouldn't have known this. But molecular techniques are picking this up more and more to show that yes, these animals are eating a substantial number of insects. Even our fruit-eating pteropods are considerably insectivorous or nectarivorous. These bats also might even be better described as nectar bats than fruit bats. The black flying fox and gray-headed flying fox will go and just sort of sit and lick flowers for uh, much of their foraging time. Um, and yes, these bats can even do things that we really would not expect. So Rosetus gypticus in Cape Town were observed actively feeding on chafer beetles. This was a very rare observation. This was like a really rare set of circumstances where there was a tree that was lit up near a hotel where Rob Barclay et al. Were staying. And they just sort of observed this by chance. So these things likely happen, but we kind of take them for granted. So when we're considering the dietary niche breadth of these animals, um, it's not just sort of what box we put them into. We really need to consider that they exist within these n dimensions. There's also records of other Rosetta species feeding on things like fish and mollusks. So like it's really sort of um, opportune when these records occur, but you need to think about this more when we're thinking about the dietary ecology of these animals. Oh, yes, frugivores eat a lot more than just fruit. And then finally, the last couple of minutes here, I'll talk about carnivores. So carnivores, there's been a lot of debate in literature of what constitutes a carnivore, carnivorous bat. There's a very good review paper that came out in 2021 called We Eat Meat. It's Gual, Suarez, and Medellin. You can check that one out. But when we think about a carnivorous bat, it's these big, charismatic animals that, yes, you guys go out and you eat other bats, you eat birds, reptiles, amphibians. But what are the bounds that we put on carnivory? So piscivory, as far as I'm concerned, we can consider this sort of an extreme or highly adapted form of carnivory. In terms of our piscivorous bats, there's a few fish-eating myotis, but Noctilio is really the one that comes to mind for me. So Noctilio leptorinus have been reported not just eating fish though, but also eating mollusks, arthropods, even other bats. So this dietary niche breadth is really sort of plastic. Trichopterosis, the fringe-lip bat, or more commonly thought of by their moniker, the frog-eating bat, because they have this sort of specialized eavesdropping behavior on Tangara frogs. Um, but this is perhaps just part of the range, or some populations do this. Whereas when there was the study by Jones et al., um, which described the diet of these frog-eating bats and found that, okay, yes, so 27% of their diet came from frogs, but also they're eating substantial number of anolis lizards, 22%, and the remaining diet was other bats or even hummingbirds. So this is a carnivorous bat, but it's eating a lot more than what it's typically described as. Other studies, when we're thinking about trichops, have estimated that the vast majority of their what they're getting are from insects. Over 80% of their diet comes from insects, including that Ingala study that I was talking about earlier. Cratopterus auricus, as I mentioned, at Laminae, they were eating plant matter, they were eating insects. And Vampirum spectrum, these guys are probably one of our true carnivores. Both of these are true carnivores, but they can be incredibly flexible in what they eat. So when we consider the dietary niches of these animals, just remember that carnivores will eat a lot more than just meat. Um, just something I wanted to leave everybody on. Just while we're constructing studies, while we're thinking about what these bats do in their environment in terms of their dietary niches, um, think beyond the trophic gill. So these work for very simple explanations, but in reality, most animals overlap in their niches. They don't fit neatly into these boxes. They share resources across these broad guilds, within these guilds, um, and we need to come up with a better definition for omnivory. We need to consider how omnivory is impacting these communities. Um, I just wanted to consider that these bats have more than a single ecological role to play as well. So these niches can be flexible, temporarily, seasonally. How these animals interact with each other in their environment changes over time as one of these other n dimensions. So yes, the n-dimensional hypervolume is alive and well. I am out of time, but I would like to thank a lot of people that have helped me along the way, various institutions I've been a part of and working with, and I'm happy to take any questions.
All right. Thank you so much, Phil. I personally I found that talk to be riveting, but also that's also my field of study. So of course I was very keen to hear everything you had to say, and very, very good job. I thought that was an incredibly informative presentation. So thank you so much.
Thank you.
And yeah, so if does anyone have any questions? We do have about like close to 10 minutes for questions here. So if anyone has any, you can feel free to put them in the chat, or you can use the raise your hand option if you like. But if I don't see any, then I will ask my question.
Okay. Should I stop sharing?
Yeah, you can stop sharing. Yeah.
Okay.
So my question's very hypothetical, which is I think you did a great job in talking about Rohit's paper, elevational gradients and how species diversity changes. That's also one of my favorite papers that's come out over the last few years. And so my question, again, kind of hypothetical, how do you think niche space, along with changing potentially an elevational gradient, do you think that latitudinally we would see something similar with species that migrate?
That is interesting. I so at latitudinal north, let's talk about since we're both in Canada.
Yeah.
Uh, we do tend to see as the tree line thins out, there's fewer bats.
Mhm.
So at the northern extremes, there's less diversity and though there's still a lot of insects. I don't know if dietary niche would be impacted, but certainly roosting availability impacts what how niche space happens at northern latitudes. Um, that would be really cool to see, especially with these bats throughout the ranges, how isotopically how isotopic niche packing occurs both at northern latitudes and at southern latitudes. But do your bats seasonally molt? Do you know where they're actually molting?
Oh man, that's the all-time question, right? It's one of the biggest problems that I ran into with that particular research was that I think there's only a single paper that came out almost 10 years ago now that describes the molting patterns for these species, and it's still pretty vague too. So
Yeah.
it's not concrete. It's not concrete.
Yeah, I mean, using other tissues would be a good way to do it, blood or patagium,
Mhm.
where there's known turnover rates. Um, is there niche packing in the tropics? Honestly, probably. But just is it a one of the things that we also did was we compared these to null models, and we found that uh, there was no significant difference from the null in terms of overlap. So there it was just sort of a mathematical quirk of increasing overlap is a result of more species. It's not an ecological phenomenon. So when there are these more diverse faunas, they do tend to overlap more. That's probably not surprising.
Yeah.
Yeah.
All right. Do we have any other questions for Dr. Oelbaum before we log off for today? And again, if you don't want to speak your question, you can put it into the chat. Okay, we've got one from Fern. You have your hand raised.
Yeah, hi, can you guys hear me okay?
Yep.
Okay, so I'm just an undergrad student. I took mammalogy last winter. This is absolutely fascinating. But I'm just curious, you're talking about like the guilds aren't super helpful except for the sanguinivorous, is it Desmodon today?
Mm.
Um, and then bringing in the whole like omnivore versus herbivore, um, where do you think the line is? Because I can see like the omnivore stuff being really helpful for large like at least in my classes, to classify different things. And like, when is it helpful to have that distinction, and when is it just totally inaccurate?
I think it's very helpful when we're talking to the public, to be able to say, look at this cool fruit bat. It's a fruit bat, and it disperses seeds. When we're actually constructing studies, uh, I think it can get in the way. So there were a few papers about the evolution of nectarivorian bats and thinking about everything is equal as a nectar bat when that's really not the case. So like a lot of the studies on nectar feeding bats revolves around or experimental studies on Glossophaga soracina or Glossophaga antillarum in some cases. Uh, and it turns out they're like kind of the worst nectar feeding bats. Like they do it half the year and they're in terms of like their physiology, they're not as adept at processing sugars as some of the other species. So when we try to equate what these bats are doing across various taxa, across various environments, it's really hard when we don't have that context. But like, if it's useful to you to talk about these bats pollinate flowers, they're nectar bats, then that's great. Um, it's just yeah, when you're sort of building up your own studies, it's good to think about it as more of a continuum or having them in being a bit more flexible in where we think of these bats in their isotopic or dietary niches.
Okay, so really just helpful for like the public. But when we're doing like actual science one day, hopefully I'll do actual science, it's just be more specifically accurate between different species.
I I think it can trip people up. If you get too stuck in thinking about what you expect an animal to be based on its dietary guild, then you're gonna eliminate a lot of possibilities. So a lot of what I was coming up against in writing that book chapter was that a lot of the records of insects in the diets of pteropods, for example, are dismissed. Like they'll mention it. It was like we found, in this pile of ejecta, we found a few discarded insects, but these were incidentally consumed, whatever. Or like, so it's being able to sort of recognize how flexible these animals are in their dietary niches, that makes the guild concept a little bit difficult to think about or to use in actual science.
That makes a lot of sense. Thank you.
And great question. And I think that this is, as you're going through your biology and ecology education, um, the concept of like, putting things into certain definitions or as Phil has been putting it into specific boxes, the further along in your education that you get, the more and more you're going to be hearing, well, we classify it as this, but there's all of these exceptions.
Yep.
And so you're just going to keep coming up against that, which are exceptions to the rule. And it's just another example of how the diversity of life doesn't like to be defined. And bats, I think, are a great example of that, which is one of the ways that makes bats so fun and exciting to study, in my humble opinion.
We're
Uh,
always learning what we don't know.
exactly. We did get a question in the chat.
Mhm.
We got it from Ramesh from Nepal. So we could see species diversity relation with niche packing. So is there any results in that niche packing is having some impact on abundance of each of the species of bat? If so, which guild? If so, which guild, depending on the higher percent of the feed, are more affected?
So, I'll use the Jamaica example that I was talking about. One of the things that I was noticing was that, for example, with the nectarivores, there's four species of nectar-feeding bat in Jamaica. Two of them are endemic to the island. Two of them are endemic to the Caribbean. Uh, when we were finding all four species together, often one of them was there were like a handful, maybe two or three, or like they were there were far less. Uh, as far as I don't have anything beyond anecdotal evidence to suggest that, there's competition involved. But certainly nectar feeding bats would be vulnerable to this, but might alleviate it through omnivory. Uh, Insect feeding bats, it's very difficult to say that there's resource limitations for insect feeding bats in the tropics. But we saw a similar effect within the family Marmopidae, where when we had all four species of Marmopids together at a site, there was more overlap among them. So they were certainly sharing resources, but abundance perhaps was never touched. Um, Yeah, I think that answers. It, it's yeah, niche packing is an interesting thing too, where the community niche space never really changed. So we were also looking to see if niche expansion was occurring. So if that sort of bubble that the whole community existed within would get bigger and that it would spread out more. But we never saw that there was no evidence to suggest that that was occurring. So the resources are in theory limiting, but so yeah, some species would be more vulnerable to greater competition than others.
Mhm. All right. Well, I would love to keep on talking about this subject, but alas, we have reached our time limit for today. So thank you everyone so much for joining us. Thank you so much to Dr. Oelbaum for joining us today and continue doing incredible research. And this video will be up on the YouTube channel here later today. So if you want to come back and revisit some of these concepts, it will be available. And all of the papers that Phil referenced in the talk, you can go back and reference those as well in the video. I know I jotted down a couple of titles myself. Yeah, so thank you everyone so much for joining us, and hopefully we will see you next time for My Field for Dummies.
Thanks for inviting me.
Thank you.
Thank you, everybody.